Go ahead.
Hi everybody, I'm Emma Franklin. I'm a senior at Class in SAS, and I'm here to talk about transposons. This relates to not only STEM, but women in science also. So transposons, also known as jumping genes or transposable elements, are mutations, are DNA strands within the body that can jump from genome to genome, creating mutations or destroying them. So humans, our DNA is composed of about 40% transposons. Less than 3% of them are active, however. This, I will explain later, is due to evolution. So let's talk about how they were founded. This was found by Dr. Barbara McClintock. Her work in maize was largely influenced by Gregor Mendel's work in pea plants and how genetics can carry over. So Dr. Barbara McClintock was inspired by him to study maize and how the chromosomes worked. So her work on chromosomes eventually turned into the coloration of maize and how it's seemingly random and there's no set goal for it, there's no purpose for it. So what she eventually found was what she called jumping genes, because they could jump from one DNA strand to another, creating mutations and randomizing these colors so much that you can hardly track where one will go next. How she did this was she injected dye into the colors in order to track where they might go. So Dr. Barbara McClintock's work was related to women in science as her work was discredited. She worked in the 1920s, and due to not only genetics were looked down upon, they were criticized, but her being a woman made this all harder for her. So she was criticized, her work was stolen by researchers, but eventually, 60 years later, she finally got the Nobel Prize in 1983, I believe. But she got the Nobel Prize and she was finally recognized for her work. So in maize, she found 80% of their DNA is composed of transposons, which are still active. So they are constantly evolving and changing and mutating. And this is greatly different from us, as we only have 3%. So let's talk about the types of transposons, which are on the left here. So there is class 1 transposons, which are retrotransposons. This makes up about 30% of the human genome, but only less than 3% of them are active. How this works, I don't have a visual model. I will for number 2. But how this works is within the DNA strand, there is one transposon. The transposon gets transcribed from the DNA into new RNA. This joins up and creates a new DNA strand, which will eventually make its way into the same genome at a different position. The thing about class 1, though, is that it never leaves the DNA strand. This is very unique and very important for class 1. Class 2, however, I do have a model of, is called the copy and paste genome. These make up less than 2% of the human genome, but are they play a larger role in genetic evolution. This is because it creates larger mutations and it can result in complete gene duplication in some cases. So how this works is you take this old strand. There is a strand of DNA, all of this is normal. The pink part is representing a transposon. So how this works is transposase grabs onto the ends of these and breaks it apart. So it takes this DNA strand and it breaks it apart from the transposon. This joins back together and makes a new DNA strand. So transposase is gripping onto the ends of these, pulls it together so that it can't join anything else, and then it finds a new DNA strand and it breaks it apart. So it breaks it apart and then it joins back together somewhere in this DNA strand. So it's copy and paste because it literally just takes it from here, puts it in here. So this is unique from class 1 because this actually leaves the DNA strand. Transposons in this way can be spurred from either natural methods like environmental factors, or they can be randomly mutated in people who are already alive, like gamma radiation. This can cause transposition to occur and great mutations to occur. So the reason why we only have less than 3% is because we don't need to change. We're in an environment where everything is the same, where we don't have to fend for ourselves to get to hunt for food and to hunt for materials. We're in a largely static environment, so there's no need for us to make changes. And if transposition were to occur in 40% of our DNA, it has more of a chance to create harmful mutations or just neither good nor bad mutations. There's a very small chance that it could create something very good. And whenever I talk about 30%, 3%, 2%, 40%, this is all kind of speculation. We can't accurately track it, but what we're inferring right now is that humans are made of about 40%. So there are specific examples from our past. Like, I have two here. The first is our ability to see in 3 colors. So our ability to see in 3 colors, 3-color vision, was found using, or studying the bones of old world primates. So we were able to look at the genes and see how this sudden mutation occurred and how it's continued over time. So our DNA, our transposons, were able to create a great mutation for us to help us survive and to help us identify certain things that will help us, that has helped us in the future. The second is our ability to digest starches. We are very different than any other animal because we have more of the ability to digest starches. We can digest potatoes and rice and more things like that. And this was due to our ability to create fire. Because we were able to break down the starches so much that we were able to eat it, this has helped us survive in times of famine.
Mr. Atwood, can you come to the office, please? Mr. Atwood, come to the office.
Because we are able to break these down, we have been able to survive over time. However, we don't need to be able to digest new starches because we already have the ones that we have. We're mass producing stuff. We don't need to create new things to help us in the future. However, implications for the future I have right here. Theoretically, this is only in theory, not in practice. In theory, if we were able to activate these transposons again, we still have them, they're just dormant. If we were able to reactivate them, we could theoretically control evolution and help us to eradicate diseases and eradicate multiple things that will kill us in the future. One example of this, on the news last week, there was a man who was recently cured from HIV. They were able to take the blood of someone who had developed a mutation against this disease, and they were able to synthesize it to where they could create a cure for him. So these mutations can help us immensely in the future. Live long lives, live good quality lives that can help us expand and help us thrive. So this is in theory, not in practice. But transposons are amazing in that they create mutations and they can bring about possibly a better future for us. All right, thanks.